Well, on the one hand, for those of you in clinical practice, a lot of what you've heard this morning should sound pretty familiar because, in fact, we use it every day. But for those of you who do cognitive science experiments, there's still something unfamiliar about what you've heard so far. We don't inject contrast agents routinely and look at the bolus as they pass, right? There are other tools that we use. As familiar as many of these steps are, there's still something fundamentally unfamiliar and one very important part of the story yet to tell. For that, I'm going to introduce our final speaker of the session, Ken Kwong. On the one hand, Ken shouldn't need much introduction to our group. Yeah, you could sit down for another minute, Ken. Just a little time. I don't want to rush this. Ken, of course, has been a fixture in the center nearly as long as I have. But, some of this, since we're talking about history, early history is worth noting. As several have mentioned, the early days of functional imaging, were performed with PET imaging. And indeed, I first met Ken when he was a fellow in the positron emission tomography group working with Dave Chesler and Nat Alpert and Jack Correa, colleagues, of course, to this day. But after his, tour in PET, he, of course, was curious to find out what was happening in the world of MR, and it was our very good fortune that he was open for that opportunity and took that opportunity. Now, you're going to hear about Ken's functional imaging experiments and, of course, where they came from and why they're so important. But for those of you who may be less familiar with the history, you might think that, that one set of experiments would, fully define, one's career. But, of course, in Ken's case, it was only a small fraction of what he did. Remember when I showed you that stroke case and we showed the perfusion image, and I said how important it was to look at that perfusion image, but in comparison to the diffusion image. Well, who was the first person to do diffusion imaging in acute stroke? It was Ken Kwong. Some of you may know about a famous paper in the CAT model from Mike Mosley, but in fact it was on a 0.6 Tesla conventional imager in the middle of a parking lot 50 feet from the White Lobby that Ken would wheel acute stroke patients from their hospital beds in the neuro ICU along with Ferdy Bonanno into that Teslacon scanner and have them sit still for an hour-long diffusion experiment because it took about 40 minutes to get a diffusion image to be able to look. And in fact, from that data, he made the very first observations of the drop in diffusion coefficient in acute stroke and did so in humans. If you've ever used the so-called FAIR technique or something analogous to that for arterial spin labeling, that was also developed by Ken Kwong right here. Ken's had this intense interest in physiological imaging of all kinds, and of course, not only has continued to make seminal contributions, but there's probably a few people here that haven't been directly as well as indirectly helped. We always know where to go for some last piece of advice. It's the guy that's always here at the lab, Ken. But for this story, it was Ken's interest in understanding hemoglobin and understanding blood and understanding magnetic susceptibility effects and how they all came together that ultimately led him to make the discoveries that brought fMRI into the realm that's probably more familiar to many of you. And for that, I'll welcome him to come to the stage and tell us all about it. Ken.
So, which computer I'm using? I guess I'm using this one. Do I just advance?
Must be this guy, so maybe that advances.
This?
Yep. Should be that.
Let me see. Okay, go back.
Okay.
All right,
and then,
fine.
I think this guy works.
All right, okay. Yeah. When I was preparing my talk, I thought, Gary wrote all these beautiful pieces on the website already about, the history of the May 9th experiment in 1991. So everybody read about it, so what else there to talk about? So I decided to give a more personal history of some of the less told story, of what led to that experiment. And the colleagues I work with that, that I learned so much from. And so came up with the title of Secret History of fMRI. So what are the secrets? First thing people usually ask me was, who was that young lady who was the subject in your May 9th experiment? And if it were 15 years ago, I would have just told you her name, but now I can't because of HIPAA, IRB regulations, so we can't do any of that thing now. So we talk about the other secret. Many other people also ask me, how did I come along to deciding that I want to do fMRI experiment? Well, I said the bright idea of linking neuronal activation to blood flow and to be imageable by MRI is entirely Jack's idea. My road to functional MRI is not because of my interest in brain functions. Jack already did it. So my main kind of single-minded interest was actually in doing perfusion imaging, trying to map brain perfusion. And when I heard about Jack's result, I know I can use the blood flow response from a neuronal activation to test any of the the perfusion mapping sequence I may come up with. So let me go to the next. Oops. Okay. Well, we already heard from Arno and Bruce about brain perfusion mapping using gadolinium, getting blood volume. But for some reason, I was very much against just a personal thing, the idea of intravascular contrast agent. So I like what sounds to me a nicer idea of using either a diffusive tracer like water, or best yet, if I can find a way to do non-contrast perfusion mapping. I had no idea how to do it. It's just a pursuit that I was going after at that time. So in around 1990, the first successful perfusion mapping experiment I did was with this somewhat funny water. It's H2O17. And normal water is H2O16. And the nice thing about H2O17, it would change the T2 of the tissue water. So when you inject it, you can see a change of a MR signal. And you can see that I circled the name of David Chesler. And that is the secret of my talk. And because David, which we normally haven't had a chance to really mention in all the history of MRI, has really been my colleague and my mentor in clarifying a lot of questions on how to do perfusion mapping, the basic concept of perfusion analysis. And so I really like to give him the credit for all the years of our working together to get to our functional MRI experiment. And just give some tidbits of the H2O17. It was very expensive, you can see. I remember, and when I was doing perfusion imaging at that time, my animal model was a rabbit. So for a rabbit, you could have to inject tens of cc. It's pretty expensive. So I had to change my model to a rat. And I went to see Bruce and say, Bruce, I need a couple of cc of this stuff, And Bruce said, yes, no problem. And this showed that his generosity of foresight, of vision, and taking up any idea completely risky. We don't know if it works or not, And so I was very happy that we get it going. And the simple lesson from this H2O17 experiment is actually being used for other upcoming experiments, including the fMRI experiment in 1991. And one of the lessons to learn is that it's actually easy to measure a change in perfusion. Because when you look at baseline perfusion, there are a lot of background signal going on that affects your MR signal. You may not be seeing what you think you are seeing, but if you have a chance to see a change in perfusion, then you know you're looking at blood flow change. And then as, again, everybody has talked about, and the CO2 hypercapnia is a very nice way to change flow. And this is a quick picture. On the right is the use of CO2. And I changed the CO2 from PCO2 from 23.5 torr to 48.5 torr. And you could see the perfusion change, which is just the kind of the slope of the decay rate, the washout rate of the HO17. Is this washout rate that gives us the perfusion of the brain. With the success of HO17 perfusion mapping behind us, but it's still a contrast agent, you know. Who wants to inject some funny water in your body? You don't know what it does 20 years from then. So besides, it's so expensive. And so when Dr. Le Bihan, also around 1990, came up with the idea, a hypothesis actually, that doing diffusion, or I should say the microcirculation in the capillaries may look like diffusion. Now, since all of us in the community at that time was very hot on doing diffusion mapping was still kind of new at that time. So we all knew how to do it, and I got very excited. Now is my ticket to doing brain perfusion without any contrast agent. So I immediately ran my favorite rabbits and applied some CO2 to it. Lo and behold, there was no change in diffusion, despite the fact we all know CO2 would change blood flow a lot. In fact, it could change 100%. So that kind of killed the idea of diffusion being able to measure perfusion, which is kind of disappointing because the whole community was really excited about it. And one extra hindsight is that, recently, some people are still trying to revive this method using diffusion measure perfusion. But the problem is that most of the diffusion sequence right now are based on EPI. And EPI could be contaminated by BO, which is an unfortunate side effect. Very hard to get rid of a BO effect. And when we were doing diffusion back in 1990s, maybe it's fortunate for us, we did not have EPI-based diffusion. Sequence wasn't written yet. So we were doing standard classical spin warp, spin echo diffusion sequence. So there's very little BO effect. So whatever we saw was probably real. Now it comes to the paper, the PNAS paper. And after second time, we failed with the diffusion to perfusion. And so I was hoping in writing in working on the PNAS paper, that the two methods I have come up by that time is one is the deoxyhemoglobin method that I learned from Keith Thorborn, and then the other method, which was actually earlier than the deoxyhemoglobin method, was the arterial spin labeling method that I learned about. And that was actually my main focus because neither of the method we know would work or not. And the deoxyhemoglobin method came much, much later because the arterial spin labeling method came first. How did I learn about arterial spin labeling? Detra and Koreski, they put up a poster in 1990 at the SMIM meeting. That shows it's good to send your postdoc to SMIM meeting because they learn a lot from there. And the paper didn't come out for 2 more years. You had to wait. That there would not have been any arterial spin labeling method for me to apply. Most of you would think of arterial spin labeling ASL as a tagging method. You tag the blood coming in, but Detra did more than that. In his paper, he described how perfusion and blood flow would change the T1 measurement of the brain tissue. And you could summarize at the bottom, basically say the T1 of brain tissue, if you apply if you add perfusion to it, you get a new T1 apparent, or 1 over T1, which is R1. That's the lesson I took from him. Now, it's kind of interesting because his method requires tagging the ASL. At that time, I wasn't quite sure how to write a new sequence with a tagging. So I wanted to use this method, but I wasn't quite ready to write a new sequence because it wasn't a commercial sequence to do it. So I thought a shortcut. What if, instead of looking at baseline perfusion, I look at a change in perfusion side. Then it became very simple by going back to his sequence. His baseline perfusion affects the baseline T1. If I do a change in perfusion, I will see a change in T1. And that's much easier to measure because now I can just measure the change in T1 if I can change the flow. And how do we change the flow? Easy, CO2, right? And of course, in addition to CO2, Jack's result already came out. And we know we can measure blood flow change with a visual stimulus. And that's what I apply. Now, in the PNAS paper, and the two time course, the top one is arterial spin labeling or T1 weighted perfusion method. The bottom is gradient echo, which is a deoxyhemoglobin sensitive method. How can you tell between the two, which one is which? Actually, pretty easy. The bottom one, the deoxyhemoglobin effect, has a post-undershoot after you turn off the light. And the reason for post-undershoot, as Joe Manaville and other folks have shown years later, is a both effect, is a blood volume effect, extra delayed blood volume result, and which is not shown at the T1 weighted perfusion method. So that's the indication that we are using to looking at two different approaches. And just, I will skip through that. That's a standby CO2 method we also use in the PSA ASP paper. And then a lot of people thought, oh, the PNAS paper only talked about visual stimulus, but we actually also did the motor cortex activation. That's finger tapping. Now, the interesting story is that after our paper was rejected by Nature, we submitted, and we were somewhat disappointed. And Jack said, we need to do something more substantial to impress the reviewer. It's not enough just to show what was shown in Science. So he thought, well, he came with the idea to link up the neuronal response and blood flow response. And the proposal he had was, well, in evoked potential, we know if you change the flickering rate of your visual stimulus, you would have different amplitude of the evoked potential. And Peter Fox also did that in his PET study already. And we wanted to show whether we could show with our MRI result, they would match the evoked potential result and the PET result. So it's all Jack's idea. I was somewhat reluctant to do it. It's an extra piece of work. But since we got rejected by Nature, we need something more substantial. And luckily, when we ran the experiment, everything fell into place, as you can see. So that was Jack's with insistence, thanks to his vision and his determination, to link the blood flow and the neuronal response together, that we got the figure in PNAS. Last, I want to pay tribute again to the key technology of fMRI, which is EPI. I didn't talk to Bruce before, but he gave a nice history of how to bring EPI to the center. But I think in the future, when Tom and Bruce are going to write a history of the MR Center, all the research accomplishment, EPI will certainly play a major role. Thank you.